All right, are you ready for chapter 8? So we are moving right along, and we finished chapter 7, which was photosynthesis, with our last one. And this one we're moving on to chapter 8, which is cellular respiration. Now with this one, once you learn photosynthesis, you'll see that a lot of processes are actually the same. So we're going to see that electron transport chain again. We're going to see the concentration of hydrogen ions on one side of the membrane. We're going to see the bringing in of nutrients and the breaking down of materials. So if you can understand photosynthesis, you can usually understand cellular respiration. Now, cellular respiration takes place in all living organisms and requires the use of the mitochondria. And so the mitochondria, it takes place in all of the major, larger organisms. And the mitochondria is the powerhouse of the cell because it's taking those sugars that were made in photosynthesis and it's turning it into that ATP energy. All right, so cellular respiration is the process that breaks down the sugar molecules, the glucose, and that was made in photosynthesis and then makes it into ATP. Oxygen is consumed during this process and carbon dioxide is a waste product. And again, remember with the same thing with cellular respiration, as with photosynthesis, is that there really aren't any waste products. The products that are created during cellular respiration can either go back and restart the process, or it can go to photosynthesis and be used in photosynthesis. So everything has a purpose. Cellular respiration is aerobic, and that means that it does require oxygen to take place. Now, there are several anaerobic methods that are like lactic acid fermentation, and these methods are just not as common as we see in the aerobic methods. Also, carbon dioxide and water are both going to be waste products. All right, so here we can see that the animals get air, or any organism that goes through this cellular respiration process is going to need oxygen. And then it goes into the mitochondria, and within the mitochondria, this process is going to take place. ADP plus phosphate is going to be combined into ATP to make that energy, and carbon dioxide and water are produced as the waste products. Now, a lot of times in our body, we will use the water for other things, but some of it does get excreted through the urinary system. All right, so the breakdown of glucose, we've got our redox reactions again. Here's our oxidation. Glucose gets oxidized into carbon dioxide, and then we have the reduction of oxygen getting reduced into that water molecule. So everything has a place. We have the same amount on this side and the same amount on that side. Nothing is lost. And energy is going to be the end product of this. Now, we also have something called NAD+. We had NADP+. You can think of the P for plants that we saw over in photosynthesis, but we have NAD+ and FAD in this reaction. Both of these are coenzymes that are needed as kind of like an energy boost for the cell, and it goes into the next process that allows the cellular respiration, like the cycle, to continue. So each NAD+ molecule is used over and over again because, again, we can add an H to this, we can take the H off, and it's just kind of like almost like a little energy source and a hydrogen acceptor that we can use for the second part of the cellular respiration process, because just like we saw with photosynthesis, there are more than one part in the cellular respiration process. Photosynthesis had the light-dependent reaction with the photosystems, and then it had the Calvin-Benson cycle. So we'll see that there are several steps in cellular respiration as well. All right, so the first step of cellular respiration, glycolysis. Glycolysis, the breakdown of glucose into two molecules of pyruvates. So before we even get to the more complicated part, that glucose molecule, because it's C6H12O6, it needs to be broken down so that the carbons aren't in that 6-ring form. And that's what glycolysis does, is it breaks down that sugar molecule into two pyruvates, and these pyruvates each go into the next phase separately. So even though we usually focus on one pyruvate during the rest of the steps, just keep in mind that this is happening to the other pyruvate that was created as well. This process does occur in the cytoplasm, and we do have just a tiny bit of ATP that is formed from this because, again, we're breaking bonds, and anytime you break bonds, energy is released. So we're not getting a whole lot of energy and ATP from this, but just a little bit. It does not utilize oxygen, so this is an anaerobic process. So our bodies can do this even without the presence of oxygen. This is really kind of a preparatory reaction where both of these molecules of pyruvates are then oxidized, and then they're going to enter the mitochondria matrix. So it starts with glycolysis, then it goes to this preparatory prep reaction, and then we're getting into the real part where the majority of the ATP is going to be created. But first, this is just kind of preparing that molecule and getting it ready. The next two steps, the citric acid cycle and the electron transport chain. These two steps are the following ones, and where most of the ATP is produced is during this electron transport chain. This is where we're going to see the electrons bounce around like that ball throwing around. This is where we are going to see the hydrogen ions getting concentrated on one side of the membrane and then going back through that ATP synthase and making that ATP. So most of the action is happening here in this electron transport chain. But the citric acid cycle does help. It's releasing 4 carbons as carbon dioxide, so this is where the majority of carbon dioxide is going to be created. And it turns twice per glucose molecule, once for each pyruvate. So remember, the glycolysis produces 2 pyruvates, and then here you're going to have those 2 pyruvates. Each one is going to enter the citric acid cycle. Produces 2 immediate ATP molecules per glucose molecule. So each one of these pyruvates is going to make another ATP. Again, bonds are being broken, but it's not very much ATP. Most of the ATP that's created, again, comes from that electron transport chain. All right, so this kind of shows the 4 different processes of cellular respiration. So again, this is happening in both plants and animals because both of them do have mitochondria, and any organism that has mitochondria is typically going to go through this process because that's really the whole point of a mitochondria. So you can see it goes into the mitochondria, and within the mitochondria we've got that electron transport chain. Here's some extra electrons that are contributed by glycolysis process, and the glucose is making it into the pyruvates. And then that goes into the electron transport chain, and then we've got these NADHs and FADHs that are also going into that chain. The citric acid cycle is a part of that as well, the preparatory phase sending it to the citric acid cycle, and eventually we're going to have that electron transport chain. So this is just kind of a flow chart showing what is happening in the glycolysis stage. 2 ATP are created, and in the citric acid stage we have 2 more ATP created. So these are 4 energies that our body can use. But really, again, it's that electron transport chain that has the vast majority of the ATP. So this would be a really good slide to come back to, as it's a really good summary to refer back to. I'm sure you have this slide in your textbook as well, but really you just need to take it step by step, and it shows you what goes into each process. And again, just like with photosynthesis, nothing is really wasted. Everything has a purpose, and then once it gets used up, it can go back to the original and start over again. Or like with the carbon dioxide and water, it can be released and used in photosynthesis instead. All right, so glycolysis this is going to break it down kind of in more detail. Before it was just an overview, so now we're kind of really getting into what's going on in each of these steps. Glycolysis occurs in the cytoplasm outside the mitochondria. So the cytoplasm, again, just surrounding the cell, this is something that isn't going to take place in the mitochondria itself. So this could take place in animals and plants that just have cytoplasm. It doesn't necessarily need to take place in organisms that have mitochondria. This is actually a method that can be used in some really tiny organisms because it does eventually produce 2 ATP. And so those 2 ATP, that's sometimes all a single-celled creature needs, is just 2 energies. And so they can just do this step and survive on this step, because if it's a small enough single-celled creature, it doesn't need anything else, which also means it's not going to need oxygen either, because if it's just doing glycolysis, then oxygen is not necessary. So glycolysis occurs in the cytoplasm outside the mitochondria. It consists of 10 reactions, each with its own enzyme, coming back to why enzymes are so important. 2 ATP are used to activate this. Remember that activation energy we talked about, that ball rolling down the hill. The ball cannot roll down the hill unless something pushes it and puts some energy into it, and enzymes lower that activation energy. So in order to get glycolysis started, we have to put 2 ATP in. 2 ATP is consumed to get this process started. This is that ball rolling down the hill. You got to put the 2 energy in. But by the time this is done, 4 ATP are created. So we get a net gain of 2 ATP. We have to use 2, we get 4, and so technically we're gaining 2 ATP each time this glycolysis happens. So 2 ATP may be enough, again, for that single-celled creature to survive. So they activate the glucose, and the glucose splits into 2 G3P molecules. There's some energy harvesting steps, oxidation, electrons, and 1 hydrogen are accepted. Here's where the NAD plus goes to an NADPH. And 4 ATP molecules are eventually produced by this thing called substrate level ATP synthesis. So it sounds kind of complicated, but basically it means that the glucose is broken down into 2 pyruvates and 4 ATP have been produced. Net gain of 2 ATP, again, because we had to use 2 to get that ball rolling. We did gain 4, but because we had to use 2, only 2 of those really count. All right, so looking at this process, we've got the glycolysis. Our input is 6-carbon glucose, C6H12O6, plus 2 NAD+, plus 2 energy, again, that energy necessary to get the reaction going, and 4 ADP and 4 phosphates. We're going to get that 2 net ATP, and this is what we have coming out. We have the 2 pyruvates. This is going to go on to the next step of cellular respiration. Now we have the 2 NADHs, and those NADHs are also going to go to the next step of cellular respiration. And then we have 4 ATP that were created with a net gain of 2. This is showing the substrate level ATP synthase, and remember, with anything that has an enzyme, we can only have certain reactions going in because only certain things will fit, and it's just showing how ATP is created from that process. Looking at glycolysis again, here's our glucose molecule, and then you put 2 energy in to activate that. It's going to have 2 energy expenditures. Eventually you are going to get these 2 G3Ps, and that is going to go into the next step where we can break it down. This is like the preparatory step I talked about before. We're getting our NADs, NADHs that can go on to a later step of cellular respiration. Just showing the different processes of how we're eventually getting to the molecule that we need to send on to the next step, those pyruvates. All right, so the pyruvate is a pivotal metabolite in cellular respiration. It's necessary, is basically what it's saying. From here, if O2 is not available, then the cell can do fermentation within the cytoplasm, and some cells do this. Yeast is a great example. Yeast can do fermentation, and if you had taken this course with a lab, we would have done a fermentation lab that demonstrates this. If oxygen is available to the cell, then the pyruvates enter the mitochondria for the aerobic portion. Fermentation will produce some energy, just not in a very high amount, and it will build up more toxic waste products. Now, our body does do a little bit of fermentation if we are exercising too much and we can't meet the oxygen demands. So if O2 is available to the cell, the pyruvates enter the mitochondria and they will go to that next step. So fermentation is an anaerobic process. We do not need oxygen for it to take place. That's what anaerobic means. And carbon dioxide or lactic acid or alcohol, those can all be produced as a waste product. So again, this is not necessarily a good thing over long term, for us anyways, because it causes a toxic buildup of acids within our body. The NADH transfers its electron to the pyruvate. Alcohol fermentation is carried out by yeast. That's where we get that ethanol smell. Lactic acid fermentation can be carried out by certain bacteria and fungi and produces lactic acid. And this is used in like products of cheese and yogurt and sauerkraut to kind of get that sour kind of taste. Other bacteria produce chemicals anaerobically, isopropanol, butyric acid, propionic acid, and acetic acid. Showing just the process of fermentation where we have those glucose molecules, and you can see kind of a cycle here of that NADH and NAD+, and then you have the byproduct of either lactic or alcohol and then carbon dioxide being produced. So some advantages of fermentation: it provides a quick ATP burst, and for our case, if we are using our muscles a lot and we can't keep up with the oxygen demands, we will switch over to lactic acid fermentation. Disadvantages is lactic and alcohol are toxic to the cells, and a buildup of these can be bad for your body. The lactic also changes the pH, and we talked about how enzymes operate better under certain conditions, and if we're screwing up those conditions and making our body more acidic, that's detrimental to the enzymes in our body. Yeast will eventually die from the alcohol they produce by fermentation if that goes on to an extreme. Efficiency of fermentation: 2 ATP produced per glucose molecule and is equivalent to 14.6 kilocalories. So if it is a small organism, it can do this and it can be just fine, but larger organisms can't really survive on just 2 ATP. We typically need more. So only 2 are produced per glucose compared to the 36 or 38 ATP produced with cellular respiration. So if oxygen is present, just do a couple more steps and you can get a more significant amount of product or ATP that your body can use. All right, so inputs and outputs of fermentation. You can see the inputs of glucose, inputs of glucose, and then the input of 2 ADP and 2 phosphates, or an output, forming an output of 2 lactic acids and 2 alcohols and 2 carbon dioxides, and a net gain of 2 ATP. Alright, so if we do have oxygen, then we move on to the next step, and this is the preparatory step that we see inside the mitochondria. This connects glycolysis to the citric acid cycle, so this is kind of an intermediate step. The end product of glycolysis, of course, is those 2 pyruvates that are going to enter into the mitochondrial matrix. Pyruvate is converted to a 2-carbon acetyl group. So this attaches to a coenzyme A and it forms something called acetyl-CoA. So acetyl-CoA is what is going to eventually go on into the mitochondria. When this happens, carbon is released and transported out, and it occurs twice per glucose molecule because remember, you end up with 2 pyruvates per 1 glucose molecule. Alright, this is showing how this reaction kind of takes place. We've got our pyruvate here, which is that 3 carbons. Remember that the original glucose started off with 6 carbons, but now we're in the 3 carbons. And we're taking those COAs, acetyl-CoAs, and we are using that NAD and making NADH, and you're eventually going to get that acetyl-CoA and the carbon dioxide as a waste product. Okay, moving on, taking a look at what the mitochondria looks like. So you're going to see that there's a lot of folding and membranes taking place in the mitochondria. And we saw a lot of membranes inside the chloroplast too. So all of this is going to take place mostly across this membrane that's all folded inside the mitochondria, just like most of photosynthesis took place across the thylakoid membrane. So the next step, after that intermediary step, It's called the citric acid cycle. This is also called the Krebs cycle, so you'll probably hear it referred to both ways. We have the Calvin-Benson cycle that was in photosynthesis, and we have the citric acid or the Krebs cycle that is found in cellular respiration. This occurs in the matrix of the mitochondria. We've got our acetyl groups, and it's used to form a 4-carbon molecule, because remember we started with 6. Those 6 still have to end up somewhere in the products. A lot of them are released, well, all of them eventually get released as carbon dioxide. NADH and FADH capture any electrons that the cell needs. And ATP is formed by something called substrate level phosphorylation. It's a really fancy term. But if you think of that Calvin cycle, how it spins around and there's the ruby P's, well, this is very similar to that. The citric acid cycle also spins around, and it kind of changes and resets itself so that the next step, which is electrons, can be produced for the electron transport chain. By the time the citric acid cycle is done, you're going to get 4 carbon dioxides, 2 ATPs, 6 NADHs, and 2 FADHs. Now, the NADHs and FADHs are those coenzymes. Those are super important, and we will see them in the electron transport chain, which comes next. And all of this happens per glucose molecule. So remember, we start off with that 1 glucose molecule, so we're going to get this for each glucose molecule. So here we can see again the glucose coming in, but glycolysis making the pyruvates. That's going in, and the electrons that are released go to the electron transport chain. But then we have the pyruvates going into the preparatory, going into the citric acid cycle, and then anything that's left is going through that electron transport chain. We've got our 2 ATP created here, and we're going to have 2 ATP created in the citric acid cycle, and then we're going to have the majority again produced in the electron transport chain. So here's how the citric acid cycle works. So we have the acetyl-CoA coming in, because that was from that intermediate, that middle step, and it's coming into our cycle, and it's going to form something called citrate. Then we have those NADHs that are formed. So here's an NAD+, and it's going to make an NADH. And then our carbon dioxide being released. Oxidation reactions producing 2 NADHs plus extra hydrogens. Again, those hydrogens coming important because they're starting to get concentrated. We have an NAD+, and then going into an NADH again. ATP is produced at this next step, where we get 1 energy from each pyruvate. FADH2 is produced, and then it kind of resets itself and goes back so the process can happen again. So we are getting NADH, FADH, and ATP from this, and the carbon dioxide is released during this step. So this is where we get our carbon dioxide. All right, so this is a good summary. Inputs and outputs. Inputs are 2 acetyl-CoA, and then 6 NAD+, 2 FAD+, and then 2 ADP and 2 phosphates. By the time we are done with the citric acid cycle, we get 4 carbon dioxides, 6 NADHs, 2 FADHs, and 2 ATP formed. So far, by just the citric acid cycle, we have a total of 4 ATP that have been created. 6 technically, okay, we've created 6 ATP, but 2 of those, again, we're not counting because we had to use 2 energy to get that ball rolling down the hill. All right, so last and arguably the most important part of cellular respiration is that electron transport chain. And with that electron transport chain, we're going to have the take place in the mitochondria and the cristae, which is the folded area. And the aerobic prokaryotics are going to have this within the plasma, within their cytoplasm, but this is the plasma membrane of the mitochondria. So a series of carrier molecules, again, if you think back to the transport chain we saw in the light-dependent reaction of photosynthesis, it used light energy to bounce around an electron to push hydrogens through the membrane. This is going to do something very similar. If you understand that process of photosynthesis, light energy going to the photosystems to push the electrons through in order to create hydrogen on one side of the membrane. If you understand that, then you can figure out the transport chain on this side. The only difference is we're not using light. What we're actually using is this NADH and this FADH that was created before. This is going to give us the energy necessary to break off those electrons and have those electrons bounce around and then push those hydrogen ions through. So instead of light, we're using NADH and FADH. Produces ATP by oxidative phosphorylation. Oxygen is the final electron acceptor, and so those hydrogen ions combine with oxygen and then it creates water as a waste product, although in our bodies and most organisms, water is not a waste product. We use the water. So the fates of the hydrogen, remember we had C6H12, so those hydrogens have to go somewhere. We've attached them to the FADHs and the NADHs, but we can use the remaining hydrogens to form those water molecules. So the carbon dioxide is formed during the Krebs cycle, and then the water molecules are formed during the electron transport chain. So hydrogen from NADH delivers enough energy to make 3 ATP. Those from FADH2 have only enough for 2 ATP. The spent hydrogens combine with oxygen to make water molecules. These coenzymes can be recycled, and this increases the efficiency of this process. It's really amazing to think how our bodies have come up with this process in order to make so much use of all of the materials. Once NADH delivers its hydrogens, it returns as NAD plus to pick up more hydrogen. So it will go back to those previous steps, the glycolysis and the the Krebs cycle, in order to pick up more hydrogens, and then it can do it again. However, hydrogens must be combined with oxygen to make water. If oxygen is not present, NADH cannot release that hydrogen ion, and it is no longer recycled back. So it has to have the oxygen present in order to pick up those extra hydrogens, and if there is no oxygen present, then it cannot be changed back to NAD+. The electron transport chain is pumping those hydrogens across that matrix and getting it into that center mitochondrial space. Again, the concentration of those hydrogens, the cell doesn't necessarily like that, because thinking back to diffusion, it likes to be balanced. It likes to have the same number of hydrogen on both sides. So if we are pumping a large amount of hydrogens to one side, that makes it unstable. And eventually, the concentration gradient is going to go from high concentration to low concentration. When this happens, it's going to go through that ATP synthase again. Those hydrogens are going to flow through, and it's going to balance it back out, and that ATP synthase is what is going to spin to make ATP out of ADP, adenosine diphosphate. This process is called chemoosmosis, where we are concentrating materials and we are trying to fix that concentration by sending those hydrogen ions through that ATP synthase. ATP moves out of the mitochondria and is used for cellular work. It can be broken down to ADP and a phosphate, and the ADP and phosphate can be used over and over again. All right, so this picture is very similar to the photosynthesis one we saw. So here we have our mitochondria membrane, and here we have the different proteins that are assisting with this process. So we have the NADH that is releasing electrons, and the electrons are getting bounced around, and the bouncing of the electrons is pushing through hydrogen. We also have hydrogen as a byproduct of this NADH when that gets released. Here we see some FADH also going into it, the hydrogen ions getting released into the mitochondrial matrix, and those electrons getting an extra boost of energy to bounce them around. So the electrons bouncing around, as those electrons are bouncing around, we have the hydrogen ions getting concentrated inside. As those hydrogen ions get concentrated, the concentration inside is greater than the concentration outside, And then we have the ATP going through into the inside of that matrix and creating that ATP molecule. So the ATP synthase is spinning around, creating that ATP energy. And it all has to deal with the bouncing of those electrons that are driving a net concentration of hydrogen ions one direction. Also, as that NADH and that FADH break apart and use up that energy that they have, the hydrogens are released and also concentrated. Everything's spinning back through the ATP synthase eventually, and that will create the energy. But those extra hydrogens, if there's extra left, will join with oxygen to create water, H2O. All right, so if we're looking at the overall yield of what we can get from one glucose molecule, from one glucose, the glycolysis process, we get 2 ATP, technically 4 ATP, but only 2 of them count. From the citric acid cycle, we can also get 2 ATP. From the electron transport chain, we can get 32 or 34, depending on how efficient the cell is and what kind of cell it is. And that gives us a grand total of about 36 ATP. The efficiency is 39%, which is not too bad considering how much energy our body needs. The rest of the energy from glucose is lost as heat. Again, we talked about how energy is not a cycle. Energy travels in one direction, and a lot of that energy is going to be lost as heat. But we are still able to get a good chunk of energy from this cellular respiration process. Okay, so here this kind of looks at the process. We're just going to put it all up here to kind of talk through it. So we've got our glycolysis that takes place in the cytoplasm. That's why this is colored different. We have energy going in, but we do have 2 ATP produced, and then we have the pyruvates. Then from the pyruvates, it goes into this next intermediate step, which makes the acetyl-CoA, and all of this is taking place inside the mitochondria. The acetyl-CoA is going to go into the citric acid cycle. Carbon dioxide is produced, more carbon dioxide is produced here, and 2 ATP overall are produced there. NADH and FADH, which are produced throughout these processes, both of these processes, are going to go into the electron transport chain. During the electron transport chain, we're going to get a variety of ATP. You can have a total of 32 or 34, and then the theoretical yield of 36 to 38 because you're combining the 4 ATP that were produced before. So 36 to 38 ATP produced per one glucose molecule. And then we can go back and put those ATP, 2 of them, into this part if needed. All right, so again, I highly recommend if you're having trouble following this, go to YouTube and watch YouTube videos about this. There are many, many good videos on YouTube that take you step by step through the photosynthesis and the cellular respiration process, and sometimes it really helps to see those molecules floating one way and molecules going the other way, and the animations do allow for a very good visual learner. So also talking about metabolism within the body. Now, your body can break down more than just glucose. We've been focusing on glucose because glucose is the most efficient, but our body is capable of breaking down proteins and lipids as well. So food sources of energy-rich molecules, typically carbohydrates, lipids, and proteins, are where we're getting the majority of our energy. And we break down these molecules, but it's called catabolism, which is the breakdown of these molecules, and it tends to be an exergonic reaction, which means it's releasing energy, which is why even animals that don't have a endothermic, making their own temperatures, they still produce a little bit of heat because energy is released during this process. Synthetic reactions is anabolism, which is the building up of molecules. So it tends to be inorganic, and this is when energy is consumed. So here we can see how different parts of the body, or different parts of nutrients, get broken down. So if we have proteins, the proteins get broken down into the amino acids because that is their monomer, again, and the polymer was the proteins. So the amino acids can be inserted into glycolysis, it can be inserted into the acetyl-CoA, or it can be inserted directly into the citric acid cycle. It depends on which type of protein was eaten. There's different kinds. So it can be inserted into different places. Now carbohydrate, of course, is going to go directly into the glycolysis, but if you have fats or lipids, then they get broken down into their glycerol heads and their fatty acid tails. The glycerol heads can go into the glycolysis process, whereas the fatty acid tails skip that process and go right to the acetyl-CoA formation to be broken down. So you can see, even though we've been talking about glycolysis or carbohydrates are the most efficient one for our bodies to use, but we can use proteins and lipids as well to make ATP. They just get inserted into different parts or different steps of that overall chain of making cellular respiration. So a lot of times this first step is skipped, and we can put some materials into glycolysis, but a lot of times it'll go into acetyl-CoA step or the citric acid step. So glucose is broken down the most efficiently. Fat breaks down into glycerol and the 3 fatty acids. Amino acids break down into carbon chains and the amino groups. And deamination is NH2 is removed, occurs in the liver. So sometimes this results in poisonous ammonia, that is NH3, that is developed. And we quickly try and convert this to urea. A lot of animals will use urea, and they will have urea or uric acid as their waste product. Birds are a great example. It's that white part you see in bird droppings. Reptiles are another good example. They also have a white part that is in their droppings, and that is urea. Different R groups from the amino acids are processed differently, and fragments enter the respiratory pathway at many different points. Again, that was some going into glycolysis, some going into acetyl-CoA, and some going directly into the citric acid cycle. Anabolism is all metabolic compounds are part of the metabolic pool. Intermediates from respiratory pathways can be used. Carbohydrates start with acetyl-CoA and basically reverse glycolysis, but in different pathways. And then fats are also used in this process. So you can do this with proteins as well. They are made up of a combination of 20 different amino acids. We'll get into that more in a different unit. And some amino acids can be synthesized by adult humans. However, other amino acids cannot be synthesized by humans. So there are some that are essential and must be present in the diet, and that's why it's important to monitor and make sure you have a healthy diet, so you're making sure you're getting these essential amino acids. If you don't have the right amino acids, then your body can't make the right proteins. So the energy organelles we visited, there are similarities between photosynthesis and cellular respiration, which we've kind of been talking about throughout this process. The use of that membrane, the chloroplast inner membrane, the thylakoid membrane, and the mitochondria, which are called cristae again. Those two membranes are both very similar for what their purposes are. The electron transport chain is similar in both photosynthesis and cellular respiration. In photosynthesis, we're using energy from the sun. In cellular respiration, we're using NADH and FADH in order to push those hydrogen ions through by the releasing of those electrons. There are different enzymes as well. In chloroplast, the stroma have the Calvin cycle enzymes, and in the mitochondria, the citric acid cycle has enzymes as well that are going to help with the reaction. So those two cycles are very similar to what they're doing. Energy flows in one direction, and eventually all energy is going to be released as heat, which isn't necessarily a form that we can use. The energy that we need is ATP, which is chemical energy. So energy flows from the sun to the chloroplast. The chloroplast makes a few ATP, but a lot of sugars, and those sugars can later be broken down by cellular respiration. We need to eat in order to get that energy, and so we'll eat plants or other animals that have eaten plants and get the energy from them. Eventually, all solar energy captured by the plants is lost, and most of it is lost in the form of heat. And a small portion of it does go to us through cellular respiration to make the ATP that our body needs. Chemicals cycle within the natural environment, so the materials can cycle, like ATP and ADP. Those get recycled. Chloroplast produces oxygen and carbohydrates. These get recycled. Carbons get turned from sugars to gases, carbon dioxide, and back again. But energy flows in one direction. Chloroplast and mitochondria allow energy flow through organisms and permit this chemical cycling. So here's a photo of the step-by-step, the two processes. This would be another one that would be great to pause at, to just look at them and make sure you're understanding. But we're seeing a lot of similarities. The Calvin cycle that we notice in photosynthesis, and then the citric acid cycle that we see in cellular respiration. The electron transport chain that is in photosynthesis versus the electron transport chain that is in cellular respiration. If you can learn one and understand one, there's just a few differences between the other. All right, and that concludes this chapter, and we will see you for the next chapter in the next video. Bye, guys. As always, if you have questions, let me know.